right, I'm going to walk through how I would analyze some data. This is going to be the process of validating virus-like sequences, trying to figure out if they're real, trying to figure out if there's errors in the assembly, trying to understand the uh, putative virus sequences that come out from a run of the Stingline Lab taxonomy pipeline. So I am logged into the AIDL NGS01 server, and right now I'm going to be looking at a data set uh, of some wheat samples that we sequenced, and these came from Punya Nachapa's lab. And the goal of this is to try to identify viruses that are infecting wheat. So yeah, I'm just going to kind of go through the process, and I'll sort of be narrating what I do. The starting point for this is the files that you can see on the screen right now that are the output of the Stingline Lab taxonomy pipeline. So what we've done in that pipeline, I'm not really going to go through, but basically that pipeline does some quality filtering, filters out host reads. So in this case, we did that by filtering all of the reads against the wheat genome. There's a good wheat genome available, so that was pretty straightforward. And then assembling all of the remaining reads, the non-wheat reads, and then mapping or trying to taxonomically categorize all of those contigs. So the contigs, you remember, are the stitched together long sequences from the short reads. And so the goal here was to try to taxonomically assess, yeah, all of those contigs. And so, and part of what the pipeline also does is it outputs files for all of the different virus-like sequences that were identified. And so you can see here, we're looking at a sample called FW23, and you can see that there's a whole bunch of virus-like sequences in this data set. So probably what I'll do is go through one or more of these and try to understand what's going on with these sequences. So just a few things before we do that. Let's have a look at what happened with the pipeline. So the way I'm going to do that is I'm going to say, I won't do shortcuts. I'll do ls minus l, let's do lrt, h, fw23 start fsq. So what this command is going to do is going to list every file that matches the pattern where it starts with fw23, and then anything, and then ends in .fsq. The -lrth option is for list. H makes the size of the files human readable. L is the long listing, and RT is sort the files in reverse order by time. So that's what that all means. So here we have, you can see 5 files. We have a beginning file, fw23_r1.fsq. That was 3.6 gigs, so pretty good. Let's see if we can figure out. Let's try this. So I did a little fastq count command to count the number of fast records in these fastq files. Fastq count-e just estimates the number, so it gives you a pretty close value, and it goes a lot faster than just counting them one by one. So you can see that, so the fw23_r1.fsq file was 3.6 gigabytes of data, and that corresponded to 10,679,615 sequences. So a pretty good sized data set there. That's a lot of reads for something like this. So that should be plenty. So then these next files correspond to the different steps in the taxonomy pipeline. So the _f is after quality filtering and removing adapter sequences. The _fu is after collapsing PCR duplicates. So U stands for unique, F stands for filtered. And then FUH adds the step of host filtering. So that's after removing host reads. So what you can see is that we started off with 10.6 million reads. After quality filtering and adapter removal, we were down to just under 10 million reads, 3.3 gigabytes of data. After the collapsing to unique reads, that actually substantially decreased the number of reads. So it went from about 9.9 million to 1.3 million. So what that tells you is that there was fair amount of PCR that was probably used to make the library. And the more cycles of PCR you do, the more PCR duplicates you have in your final library. So we remove them because it makes everything else go faster, and it's not really, uh, analyzing the exact same sequence twice does not really get you any additional new information. So because it will go faster if we remove them, and because it's not really new independent information, we remove those. So after that step, we're only down to about 13% of the starting data set. So there's a lot of PCR duplicates. And one thing that could have been done differently making the library was just to decrease the number of PCR cycles to try to get less duplication. After host filtering, that number got cut about in half, so from 1.3 million to 0.7 million, basically. So about a 50%. So only about 50% of the sequences got removed by mapping to the host to the wheat genome, which is actually kind of surprising. That's a lot of non wheat reads in the data set. So about 50% of the data was not from the host if you um, take that at face value. So then this fuhf.fastq file is what is the S here stands for singleton. So these are the reads that did not assemble into any contigs. And you can see that's about 10% of the size of the fuh.fastq file. So that means about 90% of these host filtered reads were assembled into contigs, and only about 10% of them were singletons that were not assembled into contigs. So that's sort of an overview of how the pipeline went. And like I said, the pipeline outputs all of these files, which are a whole bunch of putative virus sequences. So something you'll notice about these, so the way these are structured is this is the sort of the file, this fw23 contigs singletons.fa file is the file of contigs and singletons. And then after that, there's an underscore, and then there's this number, which is the tax ID. That actually corresponds to the tax ID of NCBI taxonomy database. So that's like an accession number for the NCBI taxonomy database. So for instance, if I take this 10847 and I go to the NCBI taxonomy database, punch that in, you can, why didn't that work? Taxonomy. Let's go to taxonomy database. So if we go here, let's search for that. You can see that that takes us to this PHYX174 genome. This is a phage. And if you click on this, you can see that its taxonomy ID is 10847. So that's like this is like the accession number for this species, the unique identifier for this species in the taxonomy database. So that's what this number is in that file. And then there's a name of that species. So here, this is Escherichia virus PHYX174. So this file will contain all of the contigs and singletons that were assigned to this species. So if we look at that by saying less, and then I'm going to paste the name of that virus, you can see there's just one contig. It's 867 bases long. The average coverage is about 2 over that contig, so not very high coverage. And this is probably a PHYX sequence. And if you may know from about Illumina sequencing that PHYX174 is a sequence that gets spiked into every Illumina run. So Illumina sells a control library that's made from PHYX DNA, and so this is probably a contaminant from that spiked in library. So we can just real quickly, I'm going to go just blast N. So this is the nucleotide blast at the NCBI website. I'm just going to paste in the sequence of that contig. Let's just have a quick look at it. It should come back as a PHYX174 sequence if everything went right. So, this is kind of funny. So, because PHYX174 is so commonly used in sequencing, and basically every Illumina sequencing run contains some PHYX sequences, what you can see here is that actually we're getting a whole bunch of random organisms showing up as blast hits, like E. coli, and that might make sense if it's a phage, but also staph sequences, these other ones, whatever they are. And these are all genome assembly. So probably and they're all hitting over 100% of that query, so 100% of the contig with 100% identity. So what this means is that probably these are errors in assembly where people included phi-X sequences inappropriately in their genome assemblies, and they got assembled supposedly as part of the genome, but really this is phi-X 174 sequence that was the spike control. So this is actually kind of, I didn't really mean to make this point, but this is an example of how genome assemblies can have contaminating sequences. Really, this is a phi-X. So here you can see phage phi-X 174 quick genome. This is really just a phi-X 174 sequence. You can see if you scroll down, there's a whole bunch of just different phi-X sequences. All these ones up at the top are all, these are all mistakes. They should be purged out of GenBank probably. Okay, so that was, if we go back here, that was this first file. There's obviously a whole bunch of files to get through. Another thing to point out about these different files is that some of them start with, context singletons.fa underscore, all this tax ID and the name of the species. Some of them have an underscore n dot fa and the name of a tax ID and the name of a virus. What you can see here is that so the difference between these ones that are .fa and underscore n.fa is the .fa ones were identified by a BLAST N type search. So these were identified at the level of sequence or nucleotide level sequence similarity. These underscore n.fa sequences are, so this file here, this fw23 underscore contigs underscore singletons underscore n.fa is the file that contains all of the contigs singletons that were not assigned at the nucleotide level. So we have these .fas. This is all the starting contigs singletons, and then everything that doesn't get assigned during that first blast n step makes it into this underscore n.fa step. So these, and these were identified by a BLAST X type search. So that's where you take nucleotide sequences, you try to find open reading frames in them, you translate those open reading frames, and then use that protein sequence to search a database of protein sequences. And from the point of identifying viruses, these are like new or more divergent viruses. So if there are sequences down in here, that could possibly be more interesting because these might correspond to previously unsequenced or more divergent viruses. Let's see. So what else do we see in here that we can talk about? So another thing to pay attention to is that there are different sizes to these files. So here we have this wheat mosaic virus file. It is this is the tax ID. That's the name of the virus, wheat mosaic virus. You can see here that this is a 28K file. So 28,000. Approximately 28,000 bases. So that's a 28,000 bases worth of sequence information. I'm not exactly sure how big the wheat mosaic virus genome is. We could look that up to see if this might correspond to a complete genome or not. That would depend on how big this virus's genome is. But you can contrast that to something like this. So human adenovirus 5. So we probably wouldn't really expect to see a human adenovirus infecting wheat, I'm guessing. So this may be a contaminant from or just a falsely assigned sequence. But in any case, you can see that the size of this file is only 200 bases. So that's quite a bit smaller than 28,000 bases. So we have a lot of sequence for this wheat mosaic virus and very little sequence for this this human adenovirus. So that's something to pay attention to too. And we see lots of different kinds of viruses. Some of them are phage. Some of them are sort of expected viruses, like these wheat viruses. Some of them might make sense. So here we have a cannabis sativa virus. It's possible that this is So this is a cannabis virus that also infects wheat. You sort of have to try to figure that out. You see other things down here, like some pineapple mealybug wilt associated virus. So this could be a new virus that's related to this pineapple mealybug wilt associated virus. Because if you remember, it's in this underscore n.fa file. So that means it was assigned at a protein level, not a nucleotide level. So it's probably a little bit more different. All right, I think that's enough sort of preamble. Let's
to walk through validating a virus sequence in one of these data sets. This is hopefully a pretty simple example. We'll find out. and I'm going to show you something I did to sort of help me keep track of what's going on. So one thing I did is I reran I created some additional tally files using the tally blast hit script that's run normally as part of the pipeline, and I ran it in a slightly different way. I'll show you what I did. So I reran the script um, for each of the data sets. I kind of this is a small little helper bash script, and I reran this tally blast hits command a couple times. And one way that I ran it here is that I used it to create these what I'm calling virus tree tally files that have just the virus-like sequences in them. Um, so I can keep, because that's what I'm ultimately keep track of. Is I'm not paying attention to bacteria or fungi or anything like that right now, just looking at viruses. So I made these virus tree tally files for all of the samples, and let's look at those. So let's look at all of them. So there's, for each of these data sets, there's 4 weak data sets. For each of those, there's 2 files, one that corresponds to the output from the blast N part of the pipeline, which is the nucleotide level similarity, and then one which corresponds to the diamond output. So yeah, so let's look at so if you look at this file, fw41 contact and singletons underscore n.fa.diamond underscore nr.virus tree tally. If you output this, this is a big table of all of the virus and virus-like sequences that were identified. And in this file, it's all of the hits, the tallies for each species have been sort of tallied up the taxonomic tree. If you hear kids yelling, sorry for that. There's kids running around because we're all working at home right now. So what I did is I outputted this file using the cat command, and let's do that one more time after clearing the screen. And then I actually just grabbed it all, copied and pasted it, and then I took it into Google's Sheets. And if you copy and paste it, because it's tabbed limited output, it actually pastes nicely into a table. And here we can see all of this information. So I'd actually done that earlier. I'll show you what that looks like. Going a little bit slow here. So I'd actually done that earlier, and I have these FW41 blast N and FW41 diamond tabs in the spreadsheet, and I actually color-coded the tallies here using some conditional formatting, which is just it's kind of nice. You can actually just click on this color scale and choose different color ranges and different values for the cutoffs for what different colors mean, and it's kind of a nice way to just real quickly visualize what's happening. And what you can see here is that it kind of really shows you the viruses that have a lot of supporting reads, they pop out, and the ones that don't kind of fall down. So here, for instance, we have P. streak virus, which is in the genus Carlovirus in the subfamily Quinviridae, and in the Beta Flexiviridae family. There's only one read from P. streak virus, and that one read gets kind of propagated up the taxonomy to this time over allele order. So I'm not really, I think, trying to dig deep into a virus that has one read is probably not really worth a lot of effort. And so I'm not going to pay attention to that. So I've sort of, in this analysis that I'm doing right now, I've sort of arbitrarily decided to only focus on sequences that have more than 20 supporting reads, which you can see right away gets rid of a lot of these lower level viruses. If you wanted to comprehensively report it, you could, but I'm not going to. You can also see things like here we have some of those 5x174 reads, so there's 36 reads mapped into 5x174. If you recall, this is a contaminant, most likely from the control Illumina library. You see a bunch of other phage. I'm not going to pay attention to the phage sequences. That's not really my goal. What you do see in this file then is, so we have 27,800 reads that map to Raboviria, which is all RNA viruses. And you can see that those 27,000 leads really come from two main species. So one of them is this wheat streak mosaic virus. The other one is this triticum mosaic virus. So we're going to actually analyze the triticum mosaic virus. There should be about 7,000 supporting reads. You can see here, this column here is median percent identity. So this is the median percent identity of the blast hits to the closest database sequence. And this is a blast N search. So this is nucleotide level similarity. So what this tells us is there's a lot of reads, and this virus here is very closely related to existing reference sequence in GenBank. So hopefully this will be a pretty easy one to analyze. So let's go look at it. Let's see here. Okay, so let's see if I can find that file. Okay, so you can see here that we have some triticum mosaic virus sequences in sample FW41. There's also some reads from that virus in other data sets, but right now let's look at this one. So let's see what we got. So I'm going to output this file. It's about 11 KB, and you can see that it's actually really nice. It's one config. That's sort of the best case scenario for a virus like this, with one genome segment. And by the way, let's do this, do that. So this, by the way, is a potivirus. It's in the Potiviridae family. These two viruses are both potiviruses, but they're in different genera and they're different species. So it looks like there's two potiviruses in the sample. And just as a reminder, potivirus. I like to go to viral zone to find out if there's a virus type of virus that I don't know that much about. Usually as a starting point, it's kind of nice to go and look to see kind of what characteristics it might have. So potiviruses are positive strand single-stranded RNA viruses, and I guess it comes from potato Y virus, and about 10 kb, so it's a single-stranded plus sense genome of about 10 kb. Our contig here is about 1,000 or 10,238 bases, so that's good. It looks like that might correspond to a full-length genome for a potivirus. Let's go see, actually. Let's see what we got. So I'm going to copy and paste this config for triticum mosaic virus. I'm going to go to my blast N page, paste it in. Let's actually just see what we get. We're going to do mega blast, so highly similar sequences. That should be just fine. Hopefully this will go pretty quickly. I'm also going to paste this sequence into a file called tmb.fa. Let's do one thing at a time. Getting a bit ahead of myself. Okay, so here we have triticum mosaic virus isolate this CO6, and we're 99.6% identical to this. It's across the whole genome. Let's look at this. So our query, which is our config, base 1 of our config corresponds to base 29 of this complete genome. So it's possible that we're missing 28 or so bases on the 5' end of the genome. And scrolling all the way down, looks like very nearly identical sequence. Scrolling down, you can see base 1020 of our query, base 10237 of our query, aligns to base 10265 of this genome. How long is this complete genome? So it is 10282. So it looks like we might be missing about 20 bases also on the 3' end of the genome. So it looks like we have possibly like near complete genome, missing 20 bases or so on each end of the genome. All right. So what I'm going to do now is use Bowtie to remap reads to the config, to this config. This config is basically the draft genome sequence for the triticum mosaic virus in our sample. And so I copied and pasted that config, and I actually copied and pasted this file called tmb.fa. So what I'm going to do now is use Bowtie build, because I want to use Bowtie to manage to it. And I'm going to run Bowtie build to take The way you run it is you give it the name of the FASTA file, and then you give it the name of the bow tie index, and this will create a new bow tie index. So I'm going to look at the files that are in my directory, and you can see that here's that tmv.fa file, and then there are these 6 new files ending in .bt2 are the bow tie index. Okay, so I'm going to run bow tie using this helper script, and I'm going to align reads from the data set BT or FW41 to that bow tie index TMV. So this will run for a little while. All right, so let's look again at the files in our directory. Here's these new files that were created by running bow tie. Let's look at the bow tie log. All right, you can see that there were, so we're running the fuh.fastq file, so that's the filtered, unique, host filtered reads against this bow tie index, so mapping to this reference sequence. So there's 214,000 reads in that fuh.fastq file, and it looks like 7,000 of them, or about 3.3%, aligned to the polyvirus reference sequence that we have. All right, so if you recall, that 7,000 is going to be pretty close to this number. We had, yeah, so that's almost exactly the same. Okay, so let's actually download the SAM file. So the SAM file contains the results of the bow tie mapping. If you look at this, you can see that there is, this is what SAM format files. This is the output of bow tie, the primary output. You can see that there's some header lines. These lines that start with the @ symbol. These header lines include things like the command line that was actually used to run bow tie, the version of bow tie, information about the reference sequences. For instance, this is the name of the reference sequence. This is the length of the reference sequence. And then all the other lines, all the rest of the lines in the file, there should be about 7,000 lines. Yeah, so there's 7,179 lines in this. Those are, there's one line of this SAM file for each read that is aligned to the reference sequence. So, and this is tab delimited, so the first thing you see here is the name of the read. This is the name of the read. This is the reference sequence that aligned to. This is the position in the reference sequence that it aligned to. So this particular read happens to align at position 1692 of the polyvirus genome. And this is the read. This is the quality of the read. There's a lot of other mapping. There's a lot of other information in this, but let's just kind of give you an idea what that looks like. So one nice thing about the SAM file is you can drag and drop that into Genius and look at the aligned reads, which is a very useful step for validating in a situation like this, validating your draft virus genome sequence. So I'm going to actually connect to the server using SFTP. So SFTP stands for Secure File Transfer Protocol. And the way you connect is, I almost did not spell that right. I misspelled something. Aha, that's the problem. I spelled AILD instead of ABL. So you basically connect like with SFTP like you would SSH. You can even do things like LS. So in CD in SFTP. So I'm going to CD to the directory where my data is. And I'm going to, so I copy that file name. I'm going to type git and then paste and then that file name, and that will download this file to my computer. So in Genius here, I have a directory for my FW41 sample. I've created a new empty folder called critical mosaic virus for this new virus. We actually need, before we drag and drop that SAM file in Genius, we need this our draft reference sequence, just so we can keep track of what's happening. Genius knows. Genius basically needs to know what the reference sequence is. It needs to know the sequence of the reference sequence. So here's my SAM file that I just downloaded. I'm going to drag and drop that into Genius. You need to have the reference sequence already in that same folder in Genius so I can figure it out. And here we have all of the reads aligned to the reference sequence. Let's go through this. So a couple things to pay attention to on this tab down here is, so I guess one thing. So here's 1 through 10,000 bases. This is the reference sequence. And then each of these little black rectangles corresponds to one read that was aligned or mapped to the genome. Here you can see a coverage chart across the whole genome. It looks like it goes from, well, it starts at 0, and then the max coverage is 229x. So somewhere along here, there's some base, it looks like over here somewhere, where there's 229 reads that map to one particular base. This would be the peak over here. And then here, this tab over here gives you some information about statistics about the mapping. So one thing you can see is that the let's see, coverage. Here we go. So yeah, the coverage goes from a minimum of 1. So there's some base somewhere in this genome that only has 1 read supporting it, and the maximum is 229x coverage. The average coverage is 99x. So on average, there's about 100 reads supporting each of it. So that's very excellent coverage. Should be plenty to get a very high quality genome. The pairwise identity between the bases and the reads in the reference sequence is overall 99.8%. So that's good. Let's see what else. So here, there's some other statistics here, like the sort of GC content of the genome. Yeah, you can look at that if you want. Zooming in, you can see this looks very good. So now, if we zoom in, you can start to see the individual bases of the reads, and you can also see highlighted bases that don't agree with the reference sequence. So up here we have the reference sequence. The reference sequence in this case is the contig that was created by the Spades assembler. And the translation underneath, although right now it doesn't know what reading frame it is in, so that doesn't make a lot of sense. You can also see all of the reads that were mapped to the genome, so these are all individual linear reads. And you can see that the bases that are different than the reference sequence are highlighted. And then where you control that, sometimes I think when you start doing this, it might not exactly look like that, but if you go over here and click on highlighting disagreements to reference, that is what you can see there. You also can see here the consensus sequence. So if there were any differences, any SNPs or variants between the reads and the reference sequence, if they were at so here we also have this consensus sequence turned on. So what we're saying is we're calling it the consensus sequence. It needs 50% support. So if there were a SNP or a consensus changing mutation in this virus compared to the reference sequence, and sometimes that happens if you're using, say, an existing GenBank reference sequence for your reference instead of your actual Spades contig, there might be a change in the consensus sequence relative to the reference sequence. Here, because we're actually already using the correct sequence, there really are no changes to the consensus sequence, which you can see if you zoom out. You can see that this top line is entirely light gray, so there are no disagreements between the consensus and the reference sequence, because otherwise they would be highlighted. So these bases, like these T's that don't match the reference, these are probably sequencing errors or errors that were introduced during library prep. They could also be legitimate low-level variants in this virus population. But this looks very good. I don't really see any problems with the assembly. It's looking very well supported. One thing I did notice before is that there was a sort of dip in coverage right here. So whenever you see like kind of an abrupt dip or change in coverage levels like that, this looks like it goes down to almost 0. Let's just zoom in and see what's going on here. Well, I mean, looking at this, it looks like the bases are well supported. It just looks like it's sort of a region of very low coverage, but there is good continuous coverage across this region. So for whatever reason, this part of the genome happens to be sort of like the low end of the distribution of coverage levels. So it's at about, so the coverage here is 15 coverage, which is Yeah, so that's just a region of low coverage, but it doesn't look like anything is necessarily wrong with it otherwise. The other thing I like to do in a situation like this is look at the ends. Um, so here you can see that this is, if you recall, I had said that based on comparison to that GenBank sequence, it looked like we were missing some bases at the 5' and 3' ends. That's pretty common for RNA viruses that were sequenced using shotgun library prep methods. It's just sort of more difficult to capture the ends of the genomes because of the way the libraries are made. So it's pretty typical to have the coverage drop off at the end, which is what you see here. You see like really high coverage out here at base about 150 of the genome, and then it decreases, and then by the time you get down here, the coverage is petered out. And this is the last, the C is the last base that's supported by our reads. So it looks like we're just going to have to accept the fact that we don't have coverage at that 5' end. Going to the 3' end, it looks like it's the same situation. We just have pretty low coverage at the end, um, just maybe even like 1x or 2x coverage. So it looks like there's a couple T's supporting this position in the genome. But otherwise, it looks good. So I'm going to call this a good reference sequence and say this is going to be the this is going to be our sort of final Triticum mosaic virus sequence for the sample FW41. It really doesn't. I'm going to copy it actually and say this is going to be Triticum virus FW41. It really doesn't get a whole lot easier than this in terms of assembling and validating virus genome sequence. It just assembled in one piece by Spades. It was just in one contig. It looks like that config I couldn't find any problems with it. It looked correct. And yeah, it looks like the only real problem we could call out here would be that we're missing a few ends at the few bases at the end of the genome. You'd have to do something like race or a different library prep method if you wanted to get sequence information for those ends. But I think it's probably coding complete, which means that probably the entire coding sequence for this is there. Let's look. So, going back to viral zone, we expect that the genome will have one big open reading frame, a big polyprotein. And it looks like here's our big polyprotein, this big open reading frame right here. So I'm just going to annotate that and add a CDS annotation, coding sequence annotation, polyprotein CDS. Now we have annotated complete genome sequence.
Now I'm going to go through another example, looking at the same virus at a Triticum mosaic virus. This is another one of these samples. This is sample FW19, and this is that same spreadsheet that I was showing you before of the table of all the virus sequences and some information about them, like how many reads aligned to them, and then how similar they were to database sequences. And we're going to, again, look at this POTI virus called Triticum mosaic virus. And here in this data set, there were only 214 reads aligning to this virus. So many fewer than that example that I just showed where there were 7,000 reads aligning to the virus. And there's, I guess, two possibilities for that. One is that the virus could be replicating at a lower level in this sample. The other option is that the data set might be a lot smaller. We can go back and look at that. Just look at the size of these data sets. So let's look at FW19. Let's look at how big that file was in the beginning of the data set. So that was 8 million reads. And FW41, where there were many more critical mosaic virus mapping reads, there was about 9.3 million. So there were more reads in the FW41 data set, but not only about 10% more reads or 12% more reads or something like that. So the size of the data set does not explain the really big difference in the number of mapping reads to Triticum mosaic virus between these data sets. If you remember from sample FW41, there were 7,000 reads mapping to that virus out of a 9 million read data set. And in this FW19 sample, there's only 214 mapping reads out of an 8 million read data set. So it looks like that in this sample, for whatever reason, this virus is just, the RNA is from this virus is present at a much lower level as measured by read mapping. So I suspect that this is a 10 kb genome. The reads are about 150 bases long, so 214 reads times 150 bases per read is going to give us about 32,000 bases of sequence information. So that means that over the entire genome, there should only be, on average, about 3x coverage, 30,000 divided by 10,000. So we're going to have pretty low coverage across the genome, and there's probably going to be gaps in coverage. There's probably going to be regions of the genome where there is 0 reads covering that part of the genome. So it's probably going to be a fragmented assembly. I highly doubt that like in the last example that we looked at in dataset FW41 that it will have assembled into one contig. I suspect there'll be just a lot of more smaller contigs plus possibly some individual singleton reads. So let's actually take a look at that. I haven't looked myself, but that's just what I'm gonna guess what happened. So let's look here at sample FW19. You can see there's also about 11 kb of sequence, so these are very similar in size, but I suspect it's going to be more fragmented. Oops, you can't just type the name of the file, you gotta, so cat. Okay, so that's exactly what happened. So in the last sample, there was one big contig, and that was our draft genome sequence. Here we have many more contigs, and they're much smaller. So there's small contigs, and then some individual reads are at the bottom. So let's actually find, so the last time in the last example, I used the SPADE assembled contig as the draft genome sequence. Here we have so many contigs, it's going to be a little bit more complicated. So to do that approach, that approach wouldn't necessarily be the best one. So I'm going to show you a different way to do it, where we're going to use an existing reference sequence as the reference sequence for mapping. So first we're going to take one of those contigs. So I just took the biggest contig, it's 1285 bases long, and I am going to blast it against the NCBI nucleotide database, against GenBank. Here again, we can see that these were on average 99.3% identical to those database sequences. So this will be another example where we have highly similar existing sequences in GenBank. It looks like there's several sequences that are all equally close, including this one on top. So let's actually click on that. So here is the accession of our a closely related GenBank sequence. So let's actually copy this accession. Show you something you can do in Genius. You can go to Genius, you can actually paste, if you go to this NCBI section of Genius, you can paste in an accession number here in the nucleotide database portion, and it will get you that sequence. So here is that same sequence that we were just looking at online, and you can see that here it is. One nice thing about getting sequences this way into Genius is that they come with their annotations. So that's kind of convenient, especially for viral genomes that are more complicated than just having a single polyprotein. One thing I noticed about this, just looking at the sequence, is that I noticed that there was some R's in there. Let's see. Does anybody see one? No, it's all swimming before my eyes. Well, I can't see one right now. Somewhere in here, there's an R. An R is an ambiguity. It stands for R is the nucleotide code for purine, so it could be either an A or a G. Here it is, R. So that's not great. Since there were equally closely related sequences from the blast hit, let's grab a different one that hopefully doesn't have any ambiguities, and we'll use that one. So let's grab this one. You can see here that this sequence is from a 2009 publication from these individuals, and they published this genome sequence in this journal back in 2009. So let's actually grab this other sequence. Hopefully this one doesn't have any ambiguities. Um, here is the, I also noticed that this is a slightly longer sequence, so it may be that this is a slightly more complete sequence. So for both those reasons, this is probably a better reference sequence to use. Um, so it's a little bit longer, and I didn't notice any ambiguous bases. Okay, so I'm going to drag and drop this. Let's actually make a new folder here for sample FW19, and I'm going to make a folder for this new virus, and then I'm going to drag this sequence into here. All right. So one thing you'll notice is that this is, because this is an RNA virus, this is actually presented as an RNA sequence. It has U's instead of T's. I'm going to convert those to DNA. So because we're virologists, we know that really in the genomic RNA, those are U's and not T's, but for the purposes of using these bioinformatics tools, sometimes it's easier to just keep everything as DNA sequences, just because the Illumina sequences that we're using are also DNA sequences. We converted the RNA to DNA in library prep. So, so here we'll be comparing DNA sequences to DNA sequences. All right, I'm going to put in, I pasted in this sequence, this reference sequence from GenBank into this file called tmv.fa. You can name it whatever you wanted, of course. And I'm going to create a bow tie index using the bow tie to build command, tmv.fa, and tmv. So I'm making the index from the reference sequence in tmv.fa, that FASTA file, and it will go into a bow tie index named tmv. So these 6 files, again, down here, these are your bow tie index. So that is what bow tie needs these files to map to this reference sequence. Now I'm going to use the run btfuh command. I did not show that before, but this is just a little helper script that will map the reads in a fuh.fastq file to some bow tie index. So the way this works is that you, this tells you how to run it. You give it a sample ID and the name of a bow tie index, and it will do this. So this $1 is the first command line argument in a bash script. It's a variable name that corresponds to that. $2 is the second command line argument to a bash script. So we're going to pass in as options to the script, the ID of our sample, and the name of the bow tie index. What it's going to do is make a new variable that takes that sample ID and adds _r1 _fuh.fastq. That'll be the name of our fastq file. And then it actually runs a second helper script. So it's a little bit of a nested bash scripts here. And it's that other bash script is called runbt. And that's going to and we're passing to that the name of this fastq file and the name of the index. The runbt script is another helper script, and it will run bowtie2. It basically just sets up a bowtie command using all these options, and then it runs it, down here. I create scripts like this just because if you find yourself running the same command repeatedly, it's kind of convenient to have a helper script like this. Okay, so now that we've made the bowtie index, let's run our helper scripts on sample FW19 on the reference, or on the bowtie index called TMV. So that's going to run for a minute. Let's see what we got here. So we have LTT is just LSNS RTH tail. That's what it's an alias for that command. So here we have, this is the output of the bowtie command. So let's look at our bowtie mapping. So in this sample we only had 12,000 host filtered reads. So that's kind of a big difference between that last sample we looked at sample FW41. This looks like, in contrast to sample FW41, where there was, I think, 200,000 some reads remaining after host filtering. Here, there's only 12,000 reads remaining after host filtering. So that's the number of reads in this fuh.fastq file. And so there's not as many non-host reads in this data set, which might account for the much smaller number of triticum mosaic virus mapping reads. So, but anyway, you can see that of these 12,000 reads in the host filtered FASTQ file, 200 of them mapped to the TMV genome, so 1.7%. And that number should match. Yep, that matches that number right here. Okay, so let's look at that mapping. So this SAM file is going to be the output of the bow tie, as we went through before. So we're going to get that. I'm still logged in with SFTP. Oh, it looks like my SFTP might have timed out. Sometimes that happens. If you don't use a SSH or SFTP command a lot, or every so often, it will kind of freeze up on you like that. That's just something that happens. So I will re-SFTP into the server. And we'll download that file. So that just took 0 seconds to download because it's a pretty small file. It's only 90 KB. It's only 200 reads. So if you recall, we made the bow tie index from this sequence. So let's go and drag this SAM file into here. And so here's the mapping of the reads across the genome. So you'll notice a couple of things compared to that last example from FW41 where there's a lot of coverage. So one thing you can see is that the coverage is much lower. So these little gray bars are where individual reads line to the genome. You can see that overall the average coverage is only about 3x, which is pretty similar to what we estimated before based on 214 150 base reads and the size of this genome. So about 3x average coverage. It ranges from 0 to 10. 10x is the highest. So like maybe right here, 1, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There's your 10x right there. So there's a base like right here where there's 10x coverage. You can see that peak up here. All right. You can also, so there's low coverage and it's variable across the genome, and you can see regions of no coverage. So it looks like, for instance, right here, there is no coverage, that we just don't happen to have any reads that come from this part of the genome. So that's too bad because it means we won't be able to get a complete genome. And that's why the assembly was in multiple smaller contigs instead of one config for the whole sequence. You can see there's other areas that don't have, that also have 0 coverage. So here, for instance, there is no coverage over this part between the end of this read and the beginning of that read. There's no coverage. And then other parts of the genome, like here, for instance. So these areas with overlapping reads are probably what correspond to the contigs that we assembled before. The other thing you can notice is that there's variance in this sequence relative to this reference genome that we used. So because the reference sequence from GenBank that we used to make the bow tie index is not exactly the same virus as what we're sequencing, there are variants, there's SNPs in the virus from sample FW19 relative to this genome. So what you can see is that like here, for instance, at this position of the genome, at position 1638 of the genome, the FW19 virus has an A, whereas the reference sequence has a G. And now the consensus has been changed to A because the consensus is the consensus of the reads, not the, so it's the consensus of all these reads, which we're defining here as 50%. So that means that more than 50% of the reads that map to this position have a base in them that's different than the reference sequence. That base will be used for the consensus sequence. Here there's a G. I also think I may have noticed, so you can see that throughout the genome there are some differences between the reads in the reference genome. So here, and you can distinguish those from like random sequencing errors. So here, this A is probably a sequencing error. The error rate of Illumina sequencing is about 0.1%. So about 1 in 1,000 bases will just by chance be incorrectly called. And that's probably what this is, although it also could be a low-level variant. So yeah, so here's another SNP. So this is a real difference between the FW19 virus and the reference sequence. So you can see how that works. You can see up here along this top line, you can see the everywhere in the genome where there's a difference in the consensus compared to the reference. So there's, not a ton, but maybe 20 or 30 differences over this 10 kb between the FW19 virus and the virus sequence that we downloaded from GenBank. So I'll show you now what we can do. We can make a new reference sequence. So I'm going to right click on that and do generate consensus sequence. So this is going to make a new consensus sequence from that SAM file from all the aligned reads. And we're going to use a 50% consensus. That's a pretty good one to use. Here we're going to actually pay a little bit of attention to some of these other options. So one thing we can say is let's call an N if coverage is less than 1. Coverage is less than 2. It's interesting, I can't go lower than 2. So yeah, that's going to be, so we're going to have a lot of N's. Anywhere there's 0x or 1x coverage in the genome, there's going to be an N in the reference sequence. And that's appropriate because we don't really know what the sequence is at that information, or at those positions in the genome. We could assume, I mean, one option you can do is you can call the reference. So you can say, use the reference sequence values when there is low coverage or quality. The problem with that is that we're making an assumption that we don't have any data to support. I mean, it's probably correct that the reference sequence bases would be the right sequence for this virus, given how similar it is to the reference sequence, but it would not be good practice to do that because we don't have data to support it. So a more conservative thing to do that we should do in this situation is to call an N when there's low coverage. So we're going to do that. We're going to make our new consensus sequence. I'm going to call it Triticum mosaic virus FW19. And let's actually even further call it partial scaffold. So scaffold means that this is a sequence that contains N's in it. So if we look around, there's going to be regions like here. So this corresponds to a region of the genome where we don't have any reads covering there. We have 0x or 1x coverage. And so there's N's in here, and the number of N's are based on the reference sequence, but we don't have sequence information. So that's good. And if you look around, you can see that there's multiple areas like that. That's curious. I wonder why there's question marks there instead of N's. I think maybe there's 0, maybe those correspond to areas where there's 1x coverage or 0x coverage. All right, so we have what here is called a scaffold, and like I said, the scaffold means it's a sequence with N's in it. So this is not great. This is, a much less desirable situation to be in than what we went through with sample FW41, where we had a complete genome sequence with lots of coverage supported. Here we only have a partial sequence. It's obviously a virus that's very closely related to these other Triticum mosaic virus sequences, but we have not been able to recover the complete genome, which is kind of a bummer. So that is where I will end that example.